Okay, we can get going. Welcome to our webinar on water quality in the Pembina River watershed. I'm Ethan Zapach. I am a watershed science technician for the Athabasca Watershed Council and very excited to help present our webinar today. We have some great speakers lined up. All right, before we get going here, I'm going to just do a land acknowledgement. As we gather online today, we acknowledge that the Pembina River watershed is located on Treaty 6 territory as well as on the Métis homeland. With this land acknowledgement, we pay our respect to the land, First Nations, Métis, Inuit, and original caregivers of the land in the Pembina River watershed. We also respect the histories, languages, and cultures of today's First Nation, Métis, and Inuit people whose presence continue to enrich the Pembina River watershed. Very grateful to be on this land, such a beautiful place that I'm sure so many of us feel connected to. So, some housekeeping items. This webinar is being recorded, so just a heads up there. And we will make that recording available to you afterwards if you want to go back and look through anything. Please don't use your video or microphone during this presentation. We will have an opportunity to ask questions after each one of the speakers presents. And if you would like to do that, please put them into the chat and we will sift through them and find some relevant questions, maybe some overlap questions if we have time to ask them. But if you don't get your question answered, no worries, because we will be sending out a survey for the end of the presentation where you will have the opportunity to drop those questions in a place and get some answers after the fact. All right. So we have Jennifer Kerr as a presenter today with Alberta Environment and Protected Areas, and they will be presenting on the Upper Athabasca Surface Water Quality Management Framework. We also have Lauren Mahon and Theo Charette with Charette Pell Poscente will be presenting about the 2-year water quality monitoring program in the Pembina River watershed, and Ken Monk with the Northern Lights Fly Fishers, a chapter of Freshwater Conservation Canada, will be presenting work on stream temperature monitoring and Arctic grayling conservation in the Pembina River watershed. And just a note that we are financially supported by Pembina Pipelines Corporation for our water quality monitoring program in the Pembina River watershed. So if we want to just take a moment and do some introductions in the chat, feel free to drop in your name, your occupation, your affiliation for being here, why you're interested in the Pembina River watershed. And we'll just do a couple minutes on here.
Awesome. Thanks, Ethan. And while you're doing that, I'll just talk a little bit about the Athabasca Watershed Council. We were founded in 2009, and we're working towards the Water for Life strategy in the Athabasca Watershed. The Athabasca Watershed itself is 24% of Alberta. It has Rocky Mountains, foothills, a little bit of parkland, boreal forest, and Canadian Shield. And those 3 Water for Life goals that we're trying to achieve are safe, secure drinking water, healthy aquatic ecosystems, and reliable quality water supplies for a sustainable economy. We are one of 11 watershed planning and advisory councils in Alberta. So we're lucky enough to have the Lesser Slave Watershed Council in our watershed as well. And we all take on different roles, foundational roles of convener and collaborator. So we like to bring people together to talk about watershed management opportunities. Recently, we just formed a Headwaters working group. I'll mention a little bit about our Pembina technical group. And we also support groups like the Lac La Biche Watershed Management Plan and stewardship groups like Baptiste and Island Lake Stewardship Group. We do monitoring reporting, so we do state of the watershed reports just to get a snapshot of what the state of the Athabasca watershed looks like. We also do policy and planning, so we make an integrated watershed management plan and try to implement that plan throughout the watershed. And we also do a lot of education and literacy. So we have an extensive children's program, and we also do a lot of outreach events like, working with agriculture producers on financial supports and riparian areas and wetlands. And that brings me to our Healthy Shorelines Initiative, where all this started. We we noticed that in the Pembina River watershed, that, it was a large watershed, about 10% of the Athabasca, and we wanted to bring groups together, the people working on the ground together to talk about watershed management. We noticed that there was a lot of restoration groups doing a lot of good riparian and wetland work. So we wanted to complement that work. And I just want to give a shout out to that group. It's made up of 13 different organizations, and they're all listed on the PowerPoint here. And we'll be talking a lot. Lauren and Theo from Charette, Pell, Poscente will be talking a lot about that water quality program that this group is overseeing for the last 2 years. And we're excited that we're able to continue the water quality program for the next 2 years. So, with that, I'd just like to hand it over to Jennifer Kerr on the Upper Athabasca Surface Water Quality Management Framework.
Thanks so much. I'm just going to get my presentation going. Confirmation that you guys can see it. Great.
Yeah, it looks awesome. Thanks, Jennifer.
No problem. Thanks so much for having me here. So I'm going to kind of pose a question to everybody. The question is, what if we could collectively manage all the different sorts of impacts from human activity on water quality in the Upper Athabasca region? So my name is Jennifer Kerr, and I'm a water quality specialist with Alberta Environment and Protected Areas. I've been a regional limnologist in resource management for the Upper Athabasca region for nearly 4 years, with over 13 years in government focusing on water quality. So today, the way I'm going to structure this talk today is I'm going to start really big and tell you about how the government manages cumulative effects of human activities at a really broad scale in the Upper Athabasca region. And then I'm going to try and narrow my focus down to talk about some of the work that the government has done on the Pembina River historically, which is the largest tributary of the Upper Athabasca region to the Athabasca River. Let's see if I can click through. So I'm going to start off with a high-level overview about the Upper Athabasca Surface Water Quality Management Framework. It's a tongue twister. So what it is, how does it work, and then I'm going to tell you show you some of the results from the first reporting cycle that was just released a couple of weeks ago, which is the first time it's been presented on publicly. And then I'm going to talk about, as I mentioned, some of the historical water quality that's been collected within the region, and how that connects to kind of the broader cumulative effects management for the Athabasca River. So what is the Surface Water Quality Management Framework? So as the name would suggest, it's a framework or an approach to monitor and manage long-term cumulative impacts of all sorts of human activities on water quality in the main stem of a region. There's currently 4 frameworks, as you can see, kind of like just screen grabs of the title page of the 4 that are present. At some point after 2014, the government stopped taking pictures on their covers, which makes me sad because there's so many beautiful photos of the Upper Athabasca region, but so be it. So it's a framework, so it's part of a kind of a broad, there's many different levels that the government uses to manage the environment within the province. There's some approaches, which on the left is like a provincial-wide approaches, which are things like the Alberta for Life, Water for Life strategy or guidelines that apply everywhere in the province. And then there's things that are regional, which is where the UAR Surface Water Quality Management Framework comes in. You can then kind of even go more granular where you start looking at things like watershed or lake management plan, and maybe someday in the future there might be a Pembina watershed management plan that might happen. But not yet. And then if you kind of go really, really into the fine details, that's where you start seeing regulation of things through approvals and conditions for pulp mills or municipal wastewater facilities or gravel mines and things like that. But we're kind of looking at that large but not province-wide scale. Currently, there are 4 frameworks for on-surface water quality, as I mentioned. The original 2 were the Lower Athabasca region and the South Saskatchewan region, which came about in 2012 and 2014, respectively. And they were kind of implemented as a regional plan. So the regional plan was implemented under Alberta Land Use Stewardship Act and has 3 different components looking at air quality, and water quantity. For the UAR and the NSR, these came about in very late December of 2022, and they there's not a regional plan yet for these two regions. There is the intent to have a regional plan, but as a result, the water quality management frameworks were implemented under a PIA, so it's a slightly different way of looking at it. And there's not currently a water quantity or air quality for these areas yet. There's two, and the remaining parts of the province, being the Peace and the Red Deer region, are in development. So the framework has got kind of 5 broad components. I'm going to go through each of these in varying levels of detail, depending on how quickly I talk. But generally, it works in the idea of setting a regional objective for that portion of the province, selecting indicators, setting management thresholds, the management response process, and then continual reporting and evaluation. So what is this regional objective that I'm talking about? So an objective is kind of like the end goal, the vision for how we want to manage water quality within the Upper Athabasca region. And for the UAR, which is Upper Athabasca region, that's to maintain current and future water uses. So maintain and protect water quality for current and future water uses. This is aligned with what's going on in the lower Athabasca region, which makes sense because water flows upstream, it keeps going downstream, so it makes sense to align them. But it does differ from other regions like the NSR and the SSR, where it's, now I'm blanking, maintain and improve, I believe is that one, which kind of makes sense too, because those regions tend to have a lot more human activity on the landscape due to populations and land use. So that's kind of the end goal. So what are these indicators that I'm talking about? So indicators, as the name would suggest, are things that indicate or tell you something about water quality. They are specific to the location. So the indicators for the Upper Athabasca region are different from areas in the South Saskatchewan region, but they're meant to be reflecting different pressures on the landscape. So they can kind of, in this table, you can see that there's broad groupings based off of like kind of classing like general indicators, nutrients, biologicals, and things like that. And some of them are pretty reflective of multiple different types of pressures, whereas some of them, like biological indicators, which are things like E. coli, wouldn't be responding to mining, but they would be a great response to agriculture or municipal wastewater treatment, things like that. So here's the list of all 29 primary indicators. I'm not going to read them off, but you can see that they generally fall within different kind of categories that was in the table before. And these were selected through a whole process, which again, I'm not going to get into, but is found within the framework document, which is publicly available on the openalberta.ca website. The UAR also has 3 what's called secondary indicators, which are parameters that didn't quite meet the criteria to have threshold set for them, and I'll get into what a threshold is, what I mean by that. But we do monitor those and we keep an eye on them, but it's not quite at the level of a primary indicator for implementing investigations and management actions to the same extent. So now we know what we're monitoring. Next question is, where are we monitoring? So this is a map of the UAR region. We have 3 different types of monitoring stations. The first is the primary station. LTRN stands for Long-Term River Network Site. So these are sites that the government monitors along the main stem of the Athabasca River that have a long history of continual monitoring at that point and can tell you a lot of information about how water quality is changing over time. So for the UAR region, we have 1 primary station, which is where we have triggers and limits set. And again, I'll explain what some of these things are. We also have a number of different other LTRN stations that are called secondary stations. So these don't have a trigger or a limit set where we're comparing against, but they do have a lot of really interesting information that we can use to see about how water quality is changing upstream of our primary station, which is Athabasca River at the town of Athabasca. And as it kind of relates to this talk today, we also have 7 secondary monitoring stations or tributary monitoring network stations that are kind of tell you information about what the water quality is on major tributaries feeding into the main Athabasca River. Specific to this talk, on the Pembina River, we have one that's located at the Water Survey of Canada gauge near Jarvie. It's not located right next to the confluence with the Athabasca River. It's a little bit upstream, but that's because there's a flow water quantity station that is co-located with, which tells a lot of really useful information. So we now know what we're looking at. We know where we're looking. So the next question is, what are we comparing to? So I keep talking about these triggers and limits. So triggers are essentially early warning signals for potential changes that might be happening within the watershed. And if it starts crossing these triggers, then it tells us we need to take a closer look. And they're based off of historical water quality data at the main stem of the river. There's two different types. There's what's called a median trigger, which is where you have 50% of historical baseline data is below that level and 50% is above. And then you have what's called a peak trigger, which is 90% of baseline data is below it. And if that wasn't complicated enough, we also have it separated individual median and peaks for different water seasons. So we've got an open water season, which is, as the name suggests, when the water is open and you get more non-point source runoff from storms and rain events and things like that. And then we also have a winter season, which is where you have ice cover and you tend to get lower flow and less runoff. So the values will change. In addition to those triggers, we also have what's called a limit, which is kind of a concentration at which we are not really, there's an increased at risk of adverse effects to aquatic organisms. And they're based off of the most stringent surface water quality guideline, which in this case is usually protection of aquatic life. Okay, so just to give you a sense of what this looks like, apologies for the slightly blurry screen grab, but this is for right out of the framework. The dissolved aluminum, you can see that it's got an open water and it's got a winter season. The limit, which is based off of that guideline, is consistent throughout the year, so it's always 50, but the median trigger and peak trigger are each a little bit different for each of the different seasons. So this is what we compare against to know if things are changing. Next step is about talking about the management response. So how does this all come together? And I'm going to go through this a little bit quick because it's all outlaid in the framework documents and I could do a whole presentation on it, but I won't. So the first step is we compare what is the water quality over the last 1 to 3 years, and we compare it to those triggers and limits to see if anything is crossing a trigger. And this is done by my colleagues in watershed sciences and using statistics. Next, if anything does cross a trigger or limit, it comes over to me, at which point I look at, is it actually trending up? And I do statistics to look at concentration trends, and I look at the influence of water quantity or flow on concentrations and trends. But I also look at things like, was it a really wet year and there was a huge flood? Or was there a spill? And that might be why there was a trigger crossing and things like that. If my findings and statistics have shown that there is indeed an increasing trend, that's where we start. Sorry, I jumped ahead of myself. That's also part of this is what we do is we assign a management level, which is basically tells us what it's like a level of 1 to 3, where 1 is basically maintain monitoring, things are not trending or haven't crossed a trigger. So we just keep looking at and keep monitoring it. 3 is a limit or one of those guidelines was exceeded and we are going to do stuff right away. And then 2 is things are trending up, but we need to figure out more information about where it's trending or why it's trending. So we open an investigation. So that's where I was jumping ahead of myself. So what is this investigation? This is where I go and I look at the data, more longer period of data or wider geographic scope of data, and try and figure out where is the water quality, what could be possible reasons for water quality indicator trending up. And we work with different stakeholders within the region. So this investigation can take into account all sorts of data, including TMN sites or like the Pembina River water quality. And it can also take into account data that's been collected by our partners, such as the Athabasca Watershed Council with their really cool Pembina water quality study. So once we know whereabouts it's going or why it's coming up, we identify ways to mitigate that or bring the trend back down. So what are these management actions that can be done to help bring things back to a more stable state or a decreasing trend within the watershed? And this, of course, is a lot easier said than done. It takes a lot of time and a lot of effort. We then would be delivering it, working with stakeholders and other people within a watershed to implement these actions. We assess how are they doing. This can take a long time, as anyone who works on large watersheds know. It's not an immediate fix. So real concerted effort. And throughout the whole process, we do a lot of communications where we're reporting on what parameters are crossing triggers or limits, and what are the management response, which is where I'm going to go next. So apologies if I'm talking a mile a minute, but happy to answer any questions after the fact. So I've talked about this whole process. So let's get to the good stuff. What is the stuff that's been found most recently? So just a couple weeks ago, the first status of the surface water quality report was released, as well as the first status of the management response for the UAR was released. And I'm going to tell you some of the most interesting parts of these studies, especially, and how it relates to the Pembina River. So again, this is mostly focused on Athabasca, the town of Athabasca. So this is the list of primary indicators. So the question is, we have these limits that we don't want crossed, that's based off of guidelines. Did any of them exceed a limit? And don't worry, I had this list. There were no limit exceedances. Yay. So that's great news. The next question is, what about those triggers? So remember, there were those median triggers and peak triggers, and there was the open water season and the ice or the winter season. So which of these, all these list of indicators exceeded or crossed a trigger? And apologies for the slightly messy slide. The next slide is a bit cleaner, but there were 6 indicators that did cross a trigger, and it depended on the season and what the trigger crossing was, but this is just the list. A little bit cleaner, I put it up in a different table, but essentially potassium had a winter median trigger crossing, sulfate had an open water median and peak. Some metals also had some trigger crossings, namely dissolved cobalt in the winter, dissolved nickel, both seasons and median trigger crossing, dissolved uranium, both seasons again, and TSS had a winter trigger crossing. But again, no limit exceedances. So the next step, I'm going to just explain it to you, and then I'm going to flash the table up. And if anyone wants to see the more detail of the table, it's all in the released report. But my next step is what I did those statistics to look at, are there trends in either the concentration-only data or the flow-adjusted data? And if there was a trend, I assigned a management level 1, 2, 3, and I stated whether or not it was closed or if it was proceeding to investigation. So that's all of this data, but the Coles notes, and the most important part, for sake of time and simplicity, is that in the case of potassium and TSS, either the trend was explained by flow or there wasn't a trend when we looked at it more closely, and as a result, these 2 indicators have been having a closed. This management response process was closed, and we're just going to go back to monitoring them year by year. But in the case of sulfate, dissolved copper, dissolved nickel, and dissolved uranium, there was either a singular positive trend or more than one positive trend, which is a direction we don't want the parameters to go. And as such, these 4 different indicators are moving to investigation. So kind of to summarize that, there are 4 new investigations that have been opened up for the UAR that are the ones I just mentioned. But to add to my fun, there's 3 additional indicators that were under investigation in the lower Athabasca region. And my colleague for the lower Athabasca region had done some really great investigative work where it's indicating that some of these parameters might be sourced from the upper Athabasca region. So as a result, he's kind of tossed those parameters upstream to me for me to kind of do additional work and implement management actions, given that it's sourced in the upper Athabasca region and just noticed in the lower Athabasca region. And the last aspect of this kind of management response report that I'm going to talk to you about is we also make recommendations to support future work. And in the case of the UAR, it's not really related to the Pembina, but we are recommending that there is enhanced monitoring to go on the main stem of the Athabasca River near Windfall. This provides a really great resolution of data between the 2 current LTRN stations at Old Entrance and Vega Ferry, and also to upgrade the TMN station on Lesser Slave River. We're also recommending to improve capture of industry collected water quality data to be able to analyze point source contributions a little bit more effectively, and to use newly developed tools such as regional environmental pressure mapping tools to assess what could be having increased environmental pressures within watersheds. So that's that. Hopefully I still seem to be going okay for time. So now that's kind of the really broad approach. So now I'm going to talk about what data does Alberta Environment have for the Pembina River, and how does that relate to potentially water quality on the main stem of the Athabasca River, which can then be potentially found through the management framework downstream through those triggers and limits that I just talked about. So kind of tying it, bringing the focus back upstream to the Pembina. So back in 2015, one of the previous limnologists in my group, Jana Tondu, had coordinated and analyzed data for what's called a winter synoptic survey. And if you're not familiar with what those terms mean, a synoptic survey means that basically, if you can imagine water as like these little parcels, you follow the same parcel of water as it floats and moves its way downstream, all the way from the headwaters, all the way down to the lower portions of the watershed, and you kind of monitor how does that water quality change, and you at the same time you also monitor various inputs. So you can monitor the tributaries that are entering into the main stem of the river, point source man-made effluents such as municipal effluent, pulp mill effluents, and they also looked at a number of different wastewaters that have approval to be released in the lower Athabasca region. However, for the sake of this talk, what I'm just going to focus in on is the Pembina River. So what things did they find was unusual about the Pembina River, or what changed? So normally the Pembina River's got an annual discharge of about 64 cubic meters per second, but because this study was done in like January, February in the middle of winter, flow was really low. So it was low flow, a lot of groundwater, no stormwater events sort of situation in this case. So they looked at water chemistry on the main stem of the river just upstream of where the Pembina came in. They looked at water quality within the Pembina itself, and then they looked at water quality on the main stem about 44 kilometers downstream. So you can kind of see what was water quality before, and did it change on the main stem after it entered, as well as what is the water quality in the river of the Pembina. So I'm going to apologize in advance. I didn't make this graph. It's a little busy, but what I'm going to, if you can try to avoid the impulse to read the map, the y-axis here is showing the concentration of a parameter, in this case dissolved oxygen. The x-axis is showing the distance as the water flows downstream. These kind of gray circles with connected lines is showing the water quality within the Athabasca River as it kind of goes down. And then the various vertical bars represent either tributaries or point sources from municipal wastewater, pulp mills, and things like that. So the main thing I'm going to draw your attention to here is just the number 10, which is the Pembina River. And then the kind of gray circles immediately above it, because that's kind of telling you what the water quality is on the main stem of the Athabasca, just around there. So for the Pembina River, I'm going to start, so now you kind of have that general gist of how these graphs work. I'm going to just give you the main talking points for a whole bunch of chemicals that they looked at. So they found that dissolved oxygen levels were generally pretty high in most of the tributaries in the upper Athabasca region, but the Pembina was quite low in this winter scenario, and it was below the short-term PAL guideline. And it also looks like it might have dropped in the Athabasca River slightly downstream of the Pembina, where the Pembina comes in as well. The Pembina River also had elevated specific conductivity relative to the Athabasca River, as you can see with that pointing with the line. And there were also a number of parameters that were elevated within the Pembina and caused an increase within the Athabasca, notably potassium, total Cal-Del nitrogen, total nitrogen, total ammonia, and total organic carbon. And if you guys are really wishing you had more time, you're welcome to take a look at the document itself. As I mentioned, just search Winter Synoptic Survey Athabasca, and it's all available on openalberta.ca. There are a number of different metals that were also found to be elevated within the Pembina River, but didn't necessarily seem to cause a change within the Athabasca, those being dissolved chromium and total and dissolved copper. So this kind of study has showed that water quality within the Pembina can be different from other tributaries within the upper Athabasca region, and that in some cases it can influence water. There is an increase that's been found downstream of within the Athabasca River. So I'm I'm going to take a moment to just kind of go back to the beginning where I'm talking about the framework. And one of the things that I didn't mention is that part of the framework is when I showed that map where it showed all the TMNs that are also monitored. One of them is that in the condition of the environment or status of surface water quality report, they also look at all of the TMNs and they say, were there any guidelines that were exceeded within a 1-year time period within any of those monitoring sites within the Upper Athabasca that Alberta Environment monitors? And if so, what parameters exceeded a guideline and how often did those guidelines exceed and at what time of year, like open water versus winter season, were those guidelines observed? So I'm going to talk to you about what they found for the Pembina River TMN site, which is again at that site that Alberta Environment monitors near Jarvie. So they found that between the period of November 1st, 2021 to October 31st, 5 indicators were found to exceed guidelines at that Pembina River site. Those being dissolved oxygen, which saw 3 very low samples, samples with low oxygen. 3 out of 5 winter samples exceeded both the acute and the chronic PAL guideline. And by exceed, I mean it was too low, not high, because dissolved oxygen, you don't want high. High is fine, low is bad. E. coli, they found 1 out of 7 open water samples exceeded the recreation guideline. And for total mercury, there was 1 sample that exceeded both the acute and chronic PAL guideline for open water. And in the case of total cobalt and total copper, there was 1 out of 7 open water samples that exceeded a chronic PAL guideline as well. So my question that I asked myself is, okay, so that's 1 year period. What is, these sites have generally been monitored since 2016 with a bit of a gap around COVID. How often are these parameters exceeding guidelines if you look at the data? So that's what I'm going to show you. So again, apologies, I'm going to take a second to just describe this graph because they're a similar pattern. Y-axis in this case is dissolved oxygen. X-axis is the date. There's a gap around COVID where sampling was suspended. And then I graphed all of the data with blue circles represent the open water, which is April through end of October. And then the black diamonds are the winter samples, so that ice cover condition. And then I also compared it to guidelines. So chronic guideline is 6.5 for dissolved oxygen, and acute guideline is 5 milligrams per liter for dissolved oxygen. So most of the time with these graphs, if it's above a guideline, that's bad. But in the case of DO, it's preferable to be above the guideline and not below it because aquatic organisms like their oxygen. So the take-home message for this kind of slide is that dissolved oxygens have historically fallen below both acute and chronic guidelines for protection of aquatic life, especially in the winter. There is this one circumstance where, I'm not sure if you can see my mouse, but there's a little blue circle that's in the open water season that's below both of the guidelines, but that sample was actually taken in April, so it's possible that it could have been more of a ice cover conditions and more representative of the winter than an open water. But during the open water season, generally, dissolved oxygen is pretty good. E. coli was found to occasionally exceed recreation and aesthetics guidelines during the open water season, generally associated with high flow events. These 2 samples here on the far right are actually, when I look at the notes from the field staff, they do mention that there's high flow, and in one case, a lot of birds upstream. So if you happen to go for a river flow to the Pembina River and there's a lot of birds, maybe don't in the water, don't stick your face in it. In the case of total mercury, concentrations of total mercury were found to occasionally exceed chronic guidelines and very rarely exceeded acute guidelines in the open water season. And these higher total mercuries were associated with high flow and turbidity levels. So it's likely associated with particulate materials and binding to sediments. In the case of total cobalt, this is a bit of a funny looking graph. The guideline for total cobalt varies with hardness, so that's why the yellow line is the guideline. The yellow dots are the guideline, and they kind of jump around a bit. So in the case of total cobalt, they occasionally exceeded PAL chronic guidelines in the open water season, and again associated with high flow and turbidity. And then the last slide that I'm going to show is talking about total copper. And this is, of course, the most complicated one. Total copper's chronic guideline is a set 7 micrograms per liter, whereas the acute guideline changes with hardness, so it kind of jumps around a bit. So in the cases of total copper, it very occasionally exceeded the chronic guideline and exceeded the PAL acute guideline on one occasion, which is this little blue circle over top of the orange dot. And that those, again, that represents 1% of all samples that exceeded the acute guideline, and generally, again, were associated with higher flow events. So in conclusion, after that whirlwind of a talk, so the UAR framework, which is the big picture stuff, it's a process to monitor, evaluate, and report on current or emerging water quality issues within the Athabasca River. The Pembina River is an important tributary to the Athabasca River, and as has been shown by that winter synoptic survey, can have water quality which differs from other tributaries within the upper Athabasca region, and downstream on the Athabasca can increase downstream of the Pembina, showing that there is connections between what's going on in tributaries and what's going on the main stem. And lastly, Alberta Environment does collect monthly water quality data at the Pembina River near the Water Survey of Canada gauging station near Jarvie, and that this kind of site, which is more closer to the confluence with the Athabasca, represents the cumulative impacts of upstream activities. So with that in mind, I believe the next speaker will be talking about some of the water quality within the Pembina River that might be occurring near that TMN site that I've been talking about, or potentially all sorts of water quality that's going on upstream of it. So zooming in even further. Thanks very much.
Thank you very much, Jennifer. That was fantastic. If anyone has any questions, feel free to put them into the chat, because Jennifer finished up with plenty of time for us to get a few questions in. Yeah, absolutely nailed it. Thank you for that. Before I introduce our next speaker.
So I see that Petra has a question that popped up in the chat. So now that we have about a decade for the lower framework and at least one reporting cycle in the upper, do you feel that the frameworks are working? And is there anything that you would do to change or improve them?
That's quite the question. I don't really focus on the lower Athabasca super much, and I've only been really wrapping my head around the frameworks over the last couple of years. So I'm kind of learning as I go, but I do think that they are a really good way of managing cumulative effects. I think that the challenge is that these things take time to change water quality, and they take time to kind of identify where things are. And there are always challenges when, stations might not be continuously monitored, or new stations come on board, and it has to have a lot of funding to keep it going for multiple stations over time. So it can be there's always it's a long-term goal, and it's a long-term sustained effort, and I think that can always be a little bit challenging when there are changes in budgets and approaches for government priorities and mandates and things like that. But I do think it's a really good way of doing it. It's more of it's not a sprint, it's a bit of a marathon.
Yeah. So I'm not sure if we have any other questions, but Ethan, if we have time, I had a little question. I know you mentioned that you were planning to upgrade some of the stations, but were there any discussions about implementing additional stations within the region at all?
Not that I'm aware of. As much as I would love to be able to be the one in charge of, and I say that, it's probably a bit of a monkey's devil. I probably wouldn't if I actually had to do it. I'm not directly involved in choosing and in developing the Alberta's MER, or monitoring, evaluation, reporting plan. That is other folks who are involved with that. So we provide a lot of feedback and our suggestions, but ultimately it's not our decision. It's based off of a lot of factors. I would love to have the Athabasca Windfall Station continuous, created as an LTRN station, just because it's an important site to know, as well as I would love to have, if it was up to me, I would have TMN sites everywhere. But unfortunately, there's a very broad provincial scope, and there's a lot of considerations that make it hard to have a lot of new stations coming on board. Fiscal and budgetary being a big one.
Do we have time for one more question, Ethan? Another one just popped up in
We
the chat.
do. I'll let it slide this time.
Okay, this is another one from Petra. Of the parameters that are trending upward, do you think we have enough info about background condition to compare them to current numbers?
Sorry, I'm just having, so do we have enough information about background conditions to compare them against current numbers? Well, I think that the background data is sufficient in order to detect trends. I think the challenge is more about refining where it's coming from due to our monitoring stations. Like, we do a great job of having a lot of stations and a lot of points within the watershed that are covered, but there's still very large areas to, they each represent a very large drainage basin. And so sometimes there's not, it would be great to have a lot more resolution within geographically to be able to really narrow it down. I think that's where it would be. If I had a magic wish list, I would be able to and then once you have a station, you also have to have it monitored continuously for about 10 years to be able to tell anything. So it's, I think, always the challenge of having enough stations for a long enough time period to be able to detect changes. But for the TMNs and the LTRN stations that we do have, I think we do have that currently.
Awesome. Thanks, Jennifer.
Yeah, and thanks everyone for your questions. Okay, well, now we can jump into Lauren's presentation. If you want to jump onto the mic there, and if you have anything to share, feel free to go in.
Kia ora, my name is Lauren. I am an environmental specialist here at CPP Environmental. And I've been leading the first couple years of this water quality investigation for the Athabasca Watershed Council Technical Advisory Group for the Pembina watershed. And yeah, I've kind of lived and breathed this project for the last couple of years, and then gone to Theo when I need some help with a few things. Do you
I'm
want to
helpful
introduce
sometimes.
yourself quickly? And then I'll share my screen as well.
Yeah, sure. Yeah, Theo, Charette. I'm the Director of Operations for CPP Environmental and aquatic ecologist. But yeah, honestly, Lauren did the vast majority of the work. So if you have any kudos to give, you should give it to her.
Okay. Trying to figure out just how to minimize. There we go. Okay.
We're not used to using Zoom. We usually use Teams, so it's like we have to relearn everything.
Yeah. Okay. So yeah, so the presentation today, hopefully I haven't put too much or too little into it, but we can always talk more about what we found already if we have some spare time, or ask any questions. But today, Theo and I will be talking to you about the first 2 years of the watershed monitoring that we've done in the Pembina River in collaboration with the AWC. So a lot of this information we're going to present will tie into what Jennifer's just presented recently. And so, yeah, we're really excited that we're kind of ready to move this project back to the AWC. The staff are really competent and confident to go forwards with setting up this long-term monitoring program. So yeah, this is kind of a summary of like the first 2 years, really. So yeah, like I said, we've been working with the AWC to set up a monitoring program with the goal of understanding water quality within the Pembina River. So we set up a monitoring program and then started implementing that for the first couple of pilot years. And then at the end of the first 2 years, we've got some interesting results already. We've just finished up a technical report, which is about to be finalized for the Technical Advisory Committee. And that will contain all of the results from the monitoring that we've done already. And then we're also, out of those first couple of years, we learned a lot of lessons, challenges, things that worked really well, things that didn't, including just how long it takes to drive from one end of the catchment to the other. So we have some recommendations for year 3 and onwards of this monitoring program, if that's going to continue. If there's any questions throughout the presentation, just pop them in the chat and then we'll get to them after we're done.
Yeah, we might, I'm not sure if we'll be able to see the
Yeah.
questions, so if the mediator could point them out to us, that would be helpful.
Okay, so just a project overview of where we're at right now. So the purpose of this program was to develop a water quality program that you can start off and then repeat year after year, pending funding and stuff like that, and personnel to do the sampling. But we wanted to design a project that you can repeat year after year, so that you're minimizing how much change is happening in terms of the sampling, and that eventually you can get up to doing the analysis that Jennifer was just referring to, things like trend analysis, longer term investigations, and the more you reduce the amount of variability in those designs, then the better quality your data is going to be. So the purpose, was to develop a repeatable program, which then we want to be able to use to inform stakeholders and partners about the water quality in the watershed. So that's the purpose of this project. And then we also developed the monitoring program itself, which is where we get things like goals and objectives. So if your goal is to determine the state of water quality in the watershed over time, there's a couple different points to that goal. One of those would be, is the water safe? So determining the state for different uses in the watershed. There's lots of different people, land uses, industry, all that sort of stuff. Recreational uses, traditional uses of surface water. So that would be objective one, is what's the state and is it safe? And then objective two is that time part of that goal. Are conditions getting better or worse over time? And that again ties back into what Jennifer was talking about earlier about those longer term sites. Right now, as of, objective one, we can start answering some of those questions. We can start seeing what might be safe for different users and what might not be safe. But objective two, we need a lot more data to be able to answer that. So that's the point of getting to a longer term, like continuous program, is that you can then answer all those objectives. And so to answer those questions, we can identify a number of indicators. This ties back to what Jennifer was talking about before, with all of those indicators and things like that that can actually tell you about the condition. I think I have a map here when I talk about. Jennifer did pull up a map earlier, but in terms of our Pembina watershed, hopefully you can see some of the labels enough. But we're talking about very, very headwaters all the way down to that site where it meets the Athabasca River down here. So the whole catchment is what we're looking at for this program. So you're looking at the watersheds in the upper area, which is things like those headwaters, right up in the mountains over here near Jasper. Sorry, my mouse has kind of disappeared a little. We're looking at the river and the tributaries where they pass through some of those small towns. So Mayerthorpe, Barhead, that sort of area. And then we're looking down in more of that prairie low-lying area near where it joins the Athabasca. So I'll show you a map a little bit later on with where we put our actual sampling sites. But this is the area that we're talking about for that monitoring program. So quite big. Let's go back here. Okay, so we can see that there's kind of steps towards developing a program such as this. We've defined our goals and objectives, so that really should be done in collaboration with community, with other stakeholders, so the TAC. We want to develop the monitoring plan itself, so what are you going to do, why, when, how, who, all that sort of stuff. And then we've done the first 2 years of our monitoring. So those are kind of the pilot years we talked about earlier. Figuring out what works and what doesn't. What are the problems that you might have to figure out before you go on and do other years? You don't really want to do 5 years of sampling and then figure out, maybe we should have changed a site or something like that a long time ago. So it's good to review what you've done already before you go and do it again the next year. So at the moment, we are here. So we have just finished doing some technical analysis and putting together recommendations for the next couple years of monitoring. So we're about here right now in the process. And yeah, you always want to be looking for improvements as you go. So here's where I'll show you the other map of where those sites are. So over the last 2 years, we have started off with 19 sites within the watershed. I might have to zoom in a little bit.
Yeah,
Here. Maybe we'll go through it step by step. So if we start up here in the headwaters, first of all, when we were selecting our sites, we really wanted to use as many locations as possible that already had some existing data. So that would be sites that perhaps were monitored for a specific project a long time ago, or they had, you know, a few samples done before the long-term site was moved somewhere else. But we wanted to use as many of those yellow locations as possible, because then you have more data as already associated with them. You can do more of that analysis about change over time. And then the pink ones here, where we've added some sites, they were selected with the stakeholders from the community group. So we've got the upper watershed in here, and then we've got some sites down here in the mid watershed. You can see the orange squares as well is where there are existing hydrometric sites. So that means that there's water level or flow discharge collected at those sites. And that's really important that we can then have water quality and river discharge collected around the same area, because it's really important for doing that analysis later on. So you can see that we started off with 19 sites, ended up with about 14 to 17. I can't quite remember when we dropped them off, but after the first year, we said that we didn't need to do a few of them, and then reduce the number of sites. And I think we're down to 14 at the moment by the end of this year. Last year was about 14 sites. Yeah, so covered a nice area of the watershed, as Jennifer was saying. We've got all the major tributaries with a site on them. So close down to the mouth of that particular river, down to that tributary where it's close to where it's contributing to the Pembina River. So that way you can see like the influence of that whole watershed of each of those tributaries. Let's go back here.
Don't water?
No, I'm okay. Do you want to talk about this for a minute? I might go get some.
Oh, okay. All right. Yeah, so basically, what we tried to do is, like you'll see with the data analysis, and you could almost put, you know, analysis under brackets, because we have, you know, 2 years of data, right? So, but the goal over the long term is to make sure that we do things the same way at the same sites, coupled with, you know, discharge data to be able to actually do proper and robust and quantitative statistical analysis. So, for example, if you wanted to do a trend analysis at whatever site, you know, you need a few years of data for that. Plus, you actually need a few years of flow or discharge data at the same time, because a lot of the variables are highly affected by flow, and you kind of want to remove that influence of flow, because you're not actually interested in that well, you are if your question is, well, what's the effect of flow on water quality? Well, in that case, you need both sets of data. But at the same time, you're actually more interested about what are the other things that affect water quality other than flow. So there's certain techniques you have to do to kind of remove that effect of flow. Does that make sense? But all of that to say is that to be able to harness the power of the data, you do need, typically for a lot of things, a longer term data set that's collected in the same way, generally. So that was really the whole focus, was to work. And I can't say enough about how collaborative this project has been, you know, between us and the Water Council, just to make sure that, so when, you know, in a month when we're gone, that the AWC could just seamlessly kind of take it over and, you know, just continue with the monitoring. And there, you know, shouldn't be really any sort of issues, right, with, you know, affecting the data. So yeah, so for now, it's kind of a starting point, and we'll talk about kind of the things and the patterns that we see. But definitely, as time goes on, you'll be able to have more and more power to be able to explore some of the questions you might have in the future.
So, like I said earlier in that steps diagram that I showed you, we've just produced our technical report, and that contains a lot of analysis about all the variables that we've been sampling for the last couple of years. In this presentation, I'm probably only going to do a couple for each kind of category, because there's a few that we'll talk a little bit more about. So, for example, these are all the results that we've collected from all those sites that were on that map over the last couple of years. So this is looking at water temperature. Down here on this graph, you can see that the sites go from the headwaters and the upper watershed down to the lower watershed. Here is that site that Jennifer was referencing back in her talk about that long-term, that tributary monitoring site. That's this one here, the Pembina River at Jarvie. You can see that there are a couple of other sites that we have that are on the main stem of the Pembina. So that's this one here, and this one here, and this one here. The rest of them are those tributaries that come in, that contribute to the Pembina. And then I've also split up on this axis up here at the top. You can just kind of reference where each of those sites are in the watershed in general. And then you'll actually look at the temperature here from all of those sites. You can see that there's actually kind of some grouping of those water temperatures. So we selected, sampled these between May-ish and end of August, I think. Over time, you want to maybe increase the range that you're collecting those samples on. But for now, we started off with just open water, trying to catch things like spring, summer, precipitation events. And then it gets a little bit more difficult once you start sampling in the autumn and the winter. So we haven't gone quite to that point yet. We stuck with the easy stuff first, low-hanging fruit.
Yeah, maybe we could mention, so what is it? We sampled, was it twice the first year?
Oh, yes. We sampled twice in 2023. We spent more time setting up the program, setting up, designing, you know, our sites, our variables that we wanted to test. And then we collected 2 samples. And then last year in 2024, we did 4 samples from these sites. Well, 3.5 really, 3.5 rounds of sampling, because in late June, I think, when areas like Jasper, there was lots of wildfires. So those upper sites have a lesser sample number than the ones in the rest of the watershed.
Because it can go down there, right?
Yeah.
Yeah.
Lots of fires everywhere, and it wasn't exactly safe to go wandering around the backcountry.
Yeah.
So there's about 6 samples here.
Okay.
Yeah.
And then the sampling, it was mostly higher to mid flows, right? Like, did we do any low flows, or I can't?
Well, that kind of goes back to what we were expecting in terms of schedule. So, for example, down here in the lower catchment, we thought that a lot of these tributaries, I didn't put any photographs of the hydrographs in here, but they seem to have like really lower flows during the late summer, based on the historical information gained from those sites with the orange squares that were on the map earlier. Those are those hydrometric sites, and so you can see over time what the general like pattern of flow is in all of those tributaries. We thought that these ones would mostly be stagnant throughout the later summer, but last year and the year before they were actually flowing, which was kind of surprising. So the other limitation of the sampling that we've done at the moment is that we wanted to get some river discharge, so through flow gauging, which you have to do through wading through the water, and we can't do that when it's unsafe either, when the flows are too high and too fast. So we want to collect data over the whole spectrum of flows from high to low, but it also has to be practically done.
Yeah, so that I guess this speaks to, you have to take sort of that long-term approach, because, you could sample each one of these tributaries and the main stem monthly, but that's going to be a heck of a lot of money. And so, or you could kind of take that sort of long-term approach that maybe you sample them 4 times a year, or maybe it's 6, whatever it is. You try to target kind of periods of, varying flows, but I would say higher and kind of mid flows. And then over a long period of time, you're going to get data from the highs and the lows. You're going to get that range of water quality information. So it's kind of using that sort of having that perspective, I guess, because it's pretty hard to justify. Like, budgets kill monitoring programs time and time again, right? If it's very expensive, it's usually, you'll go through cuts or whatever, right? And it'll be the first thing that goes, right? So you have to make sure it's manageable, from my perspective anyways.
Yeah.
So
the first couple of years that we've, even though we only have about 4 to 6 samples at each of these sites, we can already see that there are some patterns coming through. So as you increase the variability of the data, not the variability, the spectrum of data that you collect, hopefully these patterns will become more obvious, or different patterns will emerge, and then you can investigate those too. So temperature here, we can see that there's actually 3 kind of groups of temperature here throughout the watershed. They roughly correspond to those watershed boundaries. Up here is where we might find some of those more sensitive fish species up in those headwaters. But this information has a different goal to things like the Northern Lights fly fishers group that I think we're going to hear about a little bit later on. These are only spot data. They're not collecting like the diurnal variations, seasonal variations yet in all of these variables. They're just telling us about the conditions at that time of sampling throughout that time of year. But this pattern is very interesting for us to begin with. We can see, there's tributaries that might be a little bit higher, a little bit lower. Is the main stem behaving differently in terms of the temperature as you move downstream? Those are those kind of questions that you want to look at once you can actually start plotting this information. I'm going to move on because we've got lots more other stuff to get to. Here's a little bit more of an interesting graph. So this is looking at dissolved oxygen. Again, we've got the same sites, upstream to downstream. These boxes here that are kind of that gray color, those represent the lower 10th percentile. You might have to explain that a little bit, from the longer term monitoring site, so that Jarvie site. And then that box up the top here, where's my mouse gone? There we go. This box here represents where the 90th percentile, so that upper like bound of that upper group of data from that long-term site. And then the purpose of putting those on those graphs is that we can see, well, is the data that we've collected from the last 2 years, does that align with, some other data that we've had in this watershed over the past, the past few years? Are we similar or are we not? Was the last couple of years really unusual in terms of water temperature, dissolved oxygen, or was it about normal? So we can see that over the watershed, some of those results were a little bit lower. Some of them were a little bit higher up in terms of the historical data. And then, like Jennifer was talking about earlier, you can also put in guidelines. So the protection of aquatic life guideline here. So for dissolved oxygen, you really would want, over a long term, you would want your dissolved oxygen levels to be above 6.5 milligrams per liter. So you can see that most of the samples here for dissolved oxygen are. Generally, dissolved oxygen would increase as you move downstream. That's where you're looking at the pattern over time, over the watershed like that. But perhaps, there's a few sites where it's a little bit lower dissolved oxygen. So maybe you want to go and have a look at those sites in particular and see what's happening, maybe to cause those results to be a little bit lower. You can also look at tributaries and sites kind of together. So for example, up here, maybe the Lovett River is a tributary along with the Dismal and Rat Creek. They're tributaries to the main stem down at Site 7. Maybe why is the Lovett River having slightly lower dissolved oxygen than the other tributaries? There's a few other variables where down here we have the same kind of questions happening. Why is one of the tributaries behaving different from the other tributaries, even though they have the same land use, perhaps geology, other physical characteristics?
Yeah, it's important to note that the time of the year where oxygen levels are probably the lowest, I mean, not at all sites, but would be in the winter. And we didn't sample in the winter, so like the data Jennifer was showing, that was, you know, she was focusing on the winter, which is good, but we didn't do that.
Yeah. So this is just for, you know, that kind of summer data only. So we got, new change.
Oh, yeah.
Exciting. So we've got some more colorful graphs here. So there's a lot of information on this graph. So we'll just step through that here. So we're looking at total phosphorus right now. Total phosphorus concentrations are here on the left. And then we've got the same sites down here. So even if we ignore the colors for the moment, I'll get to them in a second, we're just mapping that concentration of total phosphorus from each of those samples that we took over the last couple of years at each of those sites. So automatically you can see that there's something else happening down here in the mid and the lower watershed in these sites compared to these sites up here in the upper watershed. So something is happening, something's changed for total phosphorus to become a little bit higher as we move down the catchment. A really interesting change is here in the Pell watershed. If I bring up my map again, this is where we look at things like sites along the same watershed. So between site 10 here on the Little Paddle River and then site 11 down here on the Paddle River downstream of Barhead down here, something's happening for phosphorus to be going a lot higher. These other watersheds as well, these other tributaries, they're also behaving a little bit more differently than perhaps the river site, which is in the same area. So that Jarvie site down there in the Kerr. Then we can also look at, well, are these results what we would expect from either historical data, as we talked about earlier, are they in line with what we would expect from other research? So one of the other research studies that we looked at in terms of this data is where do our nutrient levels for the Pembina sites fit into agricultural intensity in Alberta? So this study here gave us some ranges for where total phosphorus concentrations move between low agricultural intensity and the medium agricultural intensity, what ranges could you expect? And then in the high agricultural intensity, where might we expect our total phosphorus concentrations? So you can see that most of our sites appear during the summer to have phosphorus levels down in that lower agricultural intensity. Being that there's less agriculture up here in the headwaters, I would hope that would be the case. I'd be a little bit worried if we had phosphorus that was way up here in the high agriculture intensity, if there was not very much agriculture up here. So that's kind of where you start looking at things like land disturbance, land uses, even point source inputs of pollution and stuff like that. Perhaps there are some of our creeks where they're actually more impacted by phosphorus and by agriculture, perhaps than other areas in Alberta. So that's a really interesting result, is that some of these areas are perhaps a lot more impacted by agriculture, by different land uses. We can't tell you exactly why at this point. You need a lot more data to be able to do that. But some very interesting results already.
When do we have to be done by?
20 past.
20 past.
Yeah.
Okay.
25 past. Yeah. And then we also do the same thing for nitrogen here. So a different pattern. You can see we've got some increases downstream here in total phosphorus. Whereas when we look at total nitrogen, it's a lot more variable over the watershed in the last couple of years. Again, more of this information is in the report. We've talked already about more of the background level stuff as opposed to the actual results. But once that report is released, you're more than welcome to dig through that, look at all the graphs, look at all the statistical analysis. But for now, we're just kind of pointing out that some of our creeks might be, you know, a little bit more unusual in how they're reacting to perhaps different causes of, you know, varying water quality. So a lot more variation there, a little bit much to talk about right now, but different to total phosphorus, I would say. Theo, I might get you to talk about this group here.
Okay. So there's a lot going on in this slide, but let me walk you through it. So one of the ways I like to look at nutrient data is graphing it against. So this is how much disturbance there is in the watersheds of each one of those, at each one of those sites. So basically where the site is, you pick that point and then you generate the watershed upstream from that. With the idea that, which is well established in literature, that, you know, usually water quality is related to how much disturbance you have in your watershed. So then you plot that against, or you plot the total phosphorus and total nitrogen against catchment disturbance, and then you just look for patterns. And basically at the end of the day, what kind of is apparent is that you really only see high concentrations of phosphorus and nitrogen when you hit, when basically more than half of your watershed has human-related disturbances in the catchment. So 60% is kind of seems to be the threshold where you're seeing those higher concentrations, whereas lower than that, at least with the data we have, which is 2 years, 6 points of data, you never see those high concentrations. So that's interesting. Something else that's interesting is that this box here, even though you have a lot of watershed disturbance, well, you might still have lower nutrient concentration. So that might be interesting to look at in terms of what makes these systems being able to do that, right? So the Lovett River, Lobstick, Little Paddle, why are they like that? And you can kind of start looking at potentially characteristics in that watershed or in the stream itself, in the watercourse itself, that might make it like in that way. So our job is to show you patterns, show you, you know, what the data is telling us, and then I think at some point down the road, like, you know, there's more data that needs to be collected, but at some point down the road, you can start. Well, already you can start looking at, oh, okay, well, maybe if we have restoration programs, it might be worth looking at focusing on some of these types of tributaries. Although there is a word of caution in here, these are concentrations, they're not loads. This doesn't tell you anything about, so say the Paddle River, how much mass of phosphorus, how many tons of phosphorus are actually delivered to the Pembina River, which would then, you know, kind of get to some of the comments earlier about, well, you know, it appears like the Pembina River is affecting the water quality in the Athabasca River. To really be able to tease that out, you do need those load calculations, and we're not getting into that right now. We don't have enough data, but at some point down the road, that would be good to do. But, you know, I think the Paddle River in itself is valuable inherently as an aquatic system. And I think inherently, personally, this is what I believe, that there's value in making that aquatic ecosystem better and in restoring the riparian areas, or, you know, whatever, to try to make those conditions better. And inherently, it should cumulatively make the water quality in the Pembina River better and just generally aquatic ecosystems better. But that's a whole other topic of discussion, you know, on their sort of restoration management side. But for now, I think these are interesting patterns for sure.
Mhm.
Oops.
Oh, other things.
Can you do it?
So I've just pulled up a couple of our medals results. This is where I've picked these two because we have some other guidelines to see whether the state is safe, you know, for things that live in the water or for different uses, the way we're using water. So there's different guidelines that we can use to tell us whether things are safe or not. Again, we only have a couple of years of data. Over time, the more data you have, the more robust it is in terms of looking at things like these guidelines. But for now, at the time where we had some samples in the last couple of years, we can see that some of our results for total lead might even be above the guideline for like livestock watering use. So if someone's taking water straight from perhaps site 12 on the main stem of the Pembina River, maybe, I don't know, maybe we want to do a little bit more testing before we let the cows drink it or something like that. Guidelines can help inform us, you know, for different uses where, you know, might want to do a little bit more investigation. So that's an example of another use there for lead. And then here we've got another guideline for iron use. For irrigation, we can see there's a different pattern in the watershed. There's a lot more variation in our results for iron versus the last screen we looked at with the lead. A lot more variation. And again, this site 12 is doing something a little bit different to the rest of the sites. There's a couple samples which are above the guideline for use of the surface water for irrigation. So that's where, you know, you'll start looking at not only like aquatic life, not only for drinking water, but for all the different uses of water in our watershed. Those are just a couple of the medal results for lead and iron. And there's a lot more of them in that report, which we've just put together. I think we get to the more exciting stuff now. Oh, yes. So after all of those samples, all of those results from the last couple of years, what do we do now? Like we said earlier, we are not going to be so involved in this project from now. Our job was to kind of set up the project and get the Athabasca Watershed Council really competent and confident in being able to do the sampling and run this program. So over the last year, we transitioned to their staff, taking on more of the field work, organization, sampling, stuff like that. And by the end of last year, that was what was happening. I don't think I even got any phone calls by the end of the last couple of samples.
They went off on their own, right?
Yeah.
And
that was really, really cool. And that was the aim of the program, is to kind of hand it off to the community groups, right, to do this kind of monitoring. So what we've learned is that perhaps we want to continue testing for some things. Perhaps those are the things that are most valuable. We get more interesting patterns out of them, interesting results. Maybe we also want to drop a few parameters because maybe they're being tested at another site, or they're not being tested, or we could use different parameters to kind of indicate this sort of stuff. So maybe there's some things over the last couple of years that we don't need to test for anymore. Maybe they cost a lot of money, like bacteria testing, pesticides. Some of those things were not even detectable even in the springtime. So if they're not detectable when we would expect them to be, maybe we don't have to spend the money on testing them. You can always add things back in later though.
And that decision, like the pesticides and the bacteria, that decision was made last year to exclude those. Total suspended solids isn't analyzed at the Jarvie site. So we thought, well, and we do test for turbidity, and they tend to be pretty highly correlated. So we thought, well, you know, let's just be consistent with what the province is doing, and we have turbidity, so I think we're not really missing too much there. Unless the province wants to add total suspended solids, then why not, right? But it's kind of, I think being consistent is really important.
And then in terms of our sites as well, so we want to keep sampling at the sites that we've done the last year. Of those sites where there's not a hydrometric station, those orange dots on that map, we want to keep the flow gauging going. where it's safely possible to do that. So because there's no automatic system telling us what the discharge is of the river at all times, we need to go and collect that information manually. So there's 4 sites where we go and do flow gauging. And so that's so that we can have the discharge and the concentrations of those results at the same time.
Yeah, and keeping in mind that even I think with those 4 sites, we still wouldn't have discharge for all the sites, right?
There would be one that doesn't have a station and it's too deep to wade.
Yeah, so the nice thing is hydrologists are very good at using models to fill in flow gaps. So I think, I think this is good. And then, yeah, for that one site, like I said, the hydrologist, like at Alberta EPA, sorry, I keep calling it Alberta Environment because that's what it was called when I worked there. All habits die hard, but they can kind of model, fill in those data sets it's pretty straightforward to do.
And if you've got enough real data, then you can extrapolate from that in the future, all that sort of fancy stuff.
Yeah.
Fancy stuff that goes over my head. But in terms of the schedule moving forwards, what we recommend is that you focus on the spring sampling. Theoretically, the tributaries, most of them should be flowing in the spring. Some of them might keep flowing throughout the rest of the year, but based on those historic patterns of hydrology that we've seen, some of them might get to low flows during the summer, and you don't want to sample a stream that's not flowing. So maybe in the spring, focus on collecting data from all those tributaries plus the main stem. And then in the summertime, maybe some of those tributaries might be stagnant by then. So you want to focus more on collecting data from the main stem of the Pembina River. Ideally, we're going to stick to doing some open water season sampling for now, so that April, May, spring to late summer, early autumn. We also, like Theo mentioned earlier, it would be really nice if we could get some precipitation event sampling. That's kind of one of those, if it decides to rain a whole full lot, then you need to be ready to go pretty quickly.
Yeah, like within a day.
Yeah. Especially if they move through the system really, really quickly. So that's something that's a little bit trickier to achieve. But if you sample for 5 years, hopefully in that time you get some kind of samples during those events.
Yeah.
So these are the recommendations for adaptations to the monitoring program that we've made from the last couple of years. I also had a quick slide here on what else we might do next in terms of studies with the data we already have, or once we have more data, what else can we do? So there's a few interesting things here that have popped up from the results. Things like the changes from the Little Paddle to the Paddle River, what's causing those huge increases in those concentrations. But at the end of the day, is those sites actually contributing an awful lot to the Pembina River? So that's that concentration multiplied by that discharge of the river at the time of sampling gives you that actual load of that contaminant. How much is it actually contributing to the downstream sites? So even though something might be really high in concentration, if it's a really low discharge, maybe it's not actually contributing that much. Whereas if you focus perhaps on other sites where they have a lot of flow, a lot of water coming into the Pembina River, maybe they contribute more of a contaminant than some of those other sites.
When you sampled just from being there, do you recall which tributaries generally seem to flow the most?
Yes. I do. Not right now. On a map, I can tell you here.
Okay.
Yeah.
Like the
and the results, the French Creek is one that behaves really differently.
Okay.
Yeah. Paddle does. It was a little bit slow, but Paddle still does.
But it's wider, right? The Paddle?
Yeah, and that's where checking those hydrometric stations before you go out to know which ones are actually flowing and not, because then you can save a lot of time. If you're not going to go and sample, you don't have to go visit it and then an hour out of the way, right? But that's where, the last couple of years we've been sampling streams that we thought would be dry in August, and they're still moving.
I was surprised.
Yeah, so who knows? Maybe that's some other changes that are happening outside of what we're talking about.
Your highest flow came from those foothills ones.
The tributaries, you mean?
Yeah, like so. Rat and bridges were being blown.
Yeah.
Yeah. Okay.
Yeah. Yeah. Yeah, and like we have some of this information already, like we have discharge at the time of sampling, but right now we haven't quite got to the stage of mapping out those relationships, as Theo was talking about earlier, relationships between variables, how much of, concentration is controlled by how much there is flow and stuff like that.
Yeah, we did look at that, so we graphed it up, but there isn't enough data. Like it's just way too... Yeah. When you don't have enough data, you don't get the full range of, say, flows or, variable concentration to actually draw a relationship, if that makes sense. So it's just not the right time. So I think, at some point down the road.
So the goal of the program like that is, longer term, you can do stuff like this with the information that Jennifer shared earlier. They can do that where those long-term sites exist. And, we go back to the objectives of the program, right? We're trying to find out if it's safe, trying to find out the state of water quality, and then is it changing over time? So then the actual methodologies that you do for the analysis, you kind of let the data kind of lead you in terms of what you actually look at. But you're looking for things that are really interesting. You go, didn't expect that, or that's different than what I thought would be the case. And that list, there are some of those things that we found already. Again, all of this stuff is in the report, but it's been a really, really cool project, and it's been very, very satisfying being able to work on this with the staff that we have from the Athabasca Watershed Council. It's been really fun going out. I have so many cool photos, and it's just really, really awesome to go out and collaborate on a project like this with them. So that was a very quick summary of all the work that we've done in the last couple of years in the Pembina, and all of that information is going to be in the technical report, which is...
That was sent off, right? They have it.
Yes. Yes. So
thank you very much, you two. That was excellent. We have time for just one question. And I do see one in the chat. Shayla, take it away.
Yeah, so I'll read it off for you. It's from Petra, and she's just wondering, if we sample for 2 more years, including flow gauging, should we be able to then calculate the loading?
Yeah, so I mean, you can technically calculate load now, like with the data that we have. However, I wouldn't right now, but yes, I would say, I can't, did you say 5 years?
2
years. I think you could probably look at it at that time. The trick with having the data, like we have flow and chemistry, but if you don't have enough data points and you start calculating things, you might be tempted to make conclusions, but you may not, if you don't have enough data points, you don't want to do that yet. Yeah. Right. So yes, I would say, probably in a couple of years, yes. And then coupling that with, the long-term site and doing those comparisons, I think you probably could. Yeah.
I saw a question come up as well, which is something that I forgot to add to that PowerPoint. But looking at the existing data sets that we have. So for example, the riparian intactness, land use, existing water quality data itself. Once we have enough information from this program, then you go and look into the interactions of all those different factors of the watersheds themselves. What do the catchments, have in common, or what's different about them, and how might that be interacting with or producing the water quality that we're actually seeing. So there's definitely value in all of that work that's already occurred across the watershed. It just takes a bit more work to actually pair it to the technical information that we've got.
So for example, you know that graph with the quadrant, like the total phosphorus, total nitrogen, and the disturbance. That's a really good way to look at things, particularly when you don't have tons of data. But I wouldn't want to try to, say, draw a regression line, like a line between phosphorus and percent disturbance at this time, because, so say if it says there's no relationship, well, is there actually no relationship, or is it just because you don't have enough data to be able to have the spread of data to be able to tell you whether or not there was a relationship? So going back to Petra's comment, I think, do it for a couple more years, then you can start kind of looking at things a little bit more closely.
Yeah, awesome. Thank you.
Awesome. Thank you two so much. We will now jump forward and I will introduce Ken Monk. And he's going to give you a great presentation about Arctic grayling. Take it away, Ken.
Thanks. My name's Ken Monk. I'm with the Northern Lights Fly Fishers chapter of Freshwater Conservation Canada. I'd like to tell you a little bit about an Arctic grayling conservation project that we did in the Upper Pembina. And one of the main components of that was some water quality information, specifically around temperature monitoring. So Northern Lights acknowledges that the conservation works falls within Treaty 6 and Treaty 8 territories, and we acknowledge all the First Nations, Métis, and Inuit people landed who've lived, traveled, and gathered on these lands for thousands of years. A little bit about Trout Unlimited Canada and Freshwater Conservation Canada. Basically, Trout Unlimited, you're probably a little bit more familiar with. We've been around for a number of years. In November, we underwent a rebranding from Trout Unlimited to Freshwater Conservation Canada. And part of that is a reflection of the kind of work that we do. It's not just about trout. It's not about the fish. It's about the water and the ecosystem. And there's other conservation opportunities, and also to avoid some confusion with the American Trout Unlimited organization. We're a completely distinct group, and that kind of tidies up that little mess. So Freshwater Conservation Canada is the parent organization. There are a number of staff biologists there, project resources, and they do some of the bigger initiatives and major works. For example, work with the Native Trout Consortium. Northern Lights Fly Fishers is the Edmonton-based chapter. We're 100% volunteers, and projects we've done in the past include riparian work in central Alberta. We've done some grayling work, some walleye work, and we've got a proposal in to do some goldeye and mooneye work coming. So it's a good idea, especially for our chapter. The name change from Trout to Freshwater Conservation Canada makes a lot of sense because we're dealing with a lot of things that simply aren't specific to trout. Let's talk a little bit about the Arctic grayling project. Arctic grayling used to be abundant in all of basically northern Alberta. And there's a number of places that they're extirpated. They're simply not there anymore. The Upper Pembina is one of it's the lowermost natural region of Arctic grayling in the province. We started this project in 2011 and currently in 2025, and it's unlike, I realize that I shouldn't have put an end date on there because I don't think we're going to finish in 2025. Basically, to give you an idea of the location, Edson is here. So basically, straight south of Edson is the the western end of the Upper Pembina, and our project area basically goes out to about Lodgepole. So Lodgepole all the way up into the headwaters on the Pembina River. So what about all these fish that used to be here? We've got lots of anecdotal evidence and historical notes. There used to be Arctic grayling all over the place. Across the province in the 60s, we really started to notice that the populations have been declining. 70s, some had disappeared, and there's possibly only a few spawning or tributaries that still contain the Arctic grayling down in the Upper Pembina. They're very vulnerable, and they're at risk of being wiped out. So why is that? Well, it's a cumulative effects. In fact, this project was one of the primary or initial testers of the cumulative effects model. Basically, we brainstormed and came up with a number of factors that were potential contributors to this. And it's not just one, there's a number of things that can contribute to this decline in the Arctic grayling. But initially, one of the ideas was that we would try and pull levers and say, well, what happens if we pull a lever and decrease the amount of fishing or angling? What if we change some of the other things? We realized that would take years and years to try and find out if it was one or more, or which were the main contributors. So in 2016, basically, they realized that the populations were in a lot of trouble and started to pull all the levers all at once. So we ended up talking to various organizations, the people that were controlling how much water was coming out of the streams, the forestry people, the oil and gas people. We had conversations and made a few actions on a number of fronts. When I say we, I'm also talking about our major partner on this was Alberta Environment and Protected Areas. The biologists, fisheries biologists out of the Edson office, we've been working hand in hand with them for many years now. So as an overview of the entire project, we had a whole bunch of activities and some of the outcomes that are listed on the chart there. So volunteer angling, temperature data logging. I'll go into details on quite a few of these outcomes. The temperature data is one of the big ones. We did some interesting work around DNA analysis. And well, I don't have a picture about it, but we approached one of the oil and gas companies and say, we got a sediment problem at this site here. We told them about it. We told them we were concerned. And then a few weeks later, they called us up and said, all right, we've got some news for you. And a few weeks after that, the bridge was replaced and they had redone the whole sediment area and so on. I also don't have a slide on the forestry management action plans. So one of the biologists got involved with forestry management people and were able to influence some of the easement areas around some of the streams. So that was good news as well. So volunteer angling, this is one of our anglers on Rat Creek. Early on in the process, we hit a number of sites and tried to basically cover the whole upper Pembina territory. We sampled a number of streams and trying to find out where the Arctic grayling were, where they weren't, just for an initial plot. And we found that Dismal Creek and Rat Creek were two of the most likely places that we were going to find Arctic grayling based on the catch rates that we had. One of the first activities we thought we would put in a fish trap. So basically, there's steel pipes that are laid into a structure across the river, and then there's a little cage here at the bottom. And the idea is the fish are swimming up into or down upstream into this area right here, just after ice off in time for spawning. And we thought we were going to catch a bunch of fish right here in this trap. As it turned out, we didn't catch any fish, which was really a surprise. So that led us into a subsequent activity. Now, there's another picture of this trap. The idea was, let's get it in before the ice comes down. And for the most part, we got it in before the ice came down, but then a lot more ice came down. So we had some challenges at that. But still, no fish were caught. Some of our volunteers took a hike upstream on the Dismal Creek. We came across this log jam, and based on some historical imagery that we got off of satellite imagery and photo images that we had from a number of years, this log jam has been building probably since the 60s or so, and it's probably a kilometer long. And we thought, yeah, that's the problem. The fish aren't getting through because there's just no way they can navigate this massive log jam. So we took on an activity where we were looking at pit tagging the fish, basically a little transceiver that goes into the fish, much like what a veterinarian would put into a dog. And we had scanners, we put the tag in, and then when we caught a fish, we could scan it, record a serial number, and then we'd know if the fish were moving, whereabouts they had been moving. And what we found was that we were able to tag fish above and below that log jam, and we were able to catch different fish above and below, so we knew that the log jam wasn't actually a barrier. So then we had to go on and try and figure out what else was happening. Because we had all those pit tags in the stream, or in the fish in the river, we put an array across, and basically an antenna array into the stream. And as the fish swam underneath that top wire and over top of another wire that was laid across the bottom of the stream, the data was recorded, or the information about the fish passing through, the time and the fish number were recorded on a little computer. And we got some really interesting information. Most of the fish that we caught were tagged again. Some of those fish were traveling quite a bit. We caught some of them 25 kilometers, and it only took a couple of days. Some of the fish, there's one we nicknamed Gord because he kept coming back and forth through the array. So it was kind of the array must have been right in the middle of his home territory because he'd cross it several times during the period that we were monitoring that area. We have an extensive water temperature monitoring system. Basically, there's one of our volunteers. We take a data logger, encapsulate it into a little steel container, and then wrap it around a tree and put it into the bottom of the river. We did that at 23 different stations on the river. And basically, we're taking a temperature reading every 30 minutes from basically June or July until about October. So we have quite an extensive set of information data available from those temperature loggers. We also have a few air sensors so that we could do a little bit of correlation between water and air temperature. There was actually huge differences between the highs and the lows. And it was really interesting. We got a little bit of a heat map that shows us which streams were cooler and which ones were warmer. And we could tell where we were likely to find grilling based on some of those temperatures. This chart here, basically at the lower end, 7.5 degrees was a really cold temperature. Arctic grilling are most comfortable when the water is between 7.5 and 17. They get a little bit uncomfortable above 17. They're really avoiding the water when it's 20 to 25, and above 25 is really a lethal level. So this was a sort of a random year. We took the monthly average temperature. And for a number of our sites, these are some of the lower sites on the study area. Basically, Dismal Creek was nice and cool. As we get down further and further, the lower sites on the Pembina River, our July temperatures were up in that really uncomfortable range, and the fish are moving somewhere else. And these are our average temperatures. Over on the right, we've got the high temperatures. So on some of these streams, we were pushing 20, 25, 27. There's a 30-degree temperature on one of the lower loggers on the Pembina River that year. Really hot, and fish are absolutely avoiding those areas. Sorry, missed a page here. When we map it out here, we've got a bit of a heat map. So at the upper end up in this range, we've got Dismal Creek, which is almost always in that comfortable or colder, cooler range. And down at the lower end, we see a lot more red temperatures. So that's where we're getting up into that really too warm for the Arctic grayling. The greens would indicate that it's a comfortable range. So our two likely places or the two places we're finding the most Arctic grayling are right in this middle area of Dismal Creek. So we've got a nice, mostly cool temperature range. And on the middle part of middle part of Rat Creek, which was a little surprising because they get some warm water. Sorry, it's this one here. This Rat Creek site here. Mostly green, a little bit into the yellows. They're uncomfortable at times. This is August temperatures, but we did find populations in both of those too. That's what do I have there. We put up wildlife cameras so that we could monitor for non-compliant angling. Basically, there was a closure on the upper Pembina and tributaries upstream from Lodgepole area. So there's no fishing allowed in that area, and especially at these major crossings. This is a site on the Pembina River. There's signage up there that says no fishing, and on our wildlife cameras, we can see people that are clearly fishing. In fact, I approached one guy. We came up to do some maintenance on a site, and I said, there's no fishing allowed here. And he was very apologetic and said, I didn't know. Well, obviously, he didn't read the regulations or any of the signs, or he was just choosing to ignore it. But we've got 2 or 3 anglers standing in this spot in the river doing some angling. We passed that along to the folks at Fish and Wildlife, or the conservation officers, and really, these were taking pictures at 15-minute intervals through the years. We got tens of thousands of images, and all we could do was report them at the end of the year and say, all right, there were people fishing there, and the next year they would go out and do a little bit more. If they had the opportunity, they would do a little bit more enforcement at those sites. Overall, though, quite a low number of those types of incidents. We also had a program where we did some aerial photography. So we had a drone that went up, and we were looking to do inventory of sites, trying to figure out, where there was sediment, Where in this particular case, we've got images of UHV activity. So basically the quads or whatever devices were riding through the stream here. If you look closely, you can see, oh, sorry, the mouse on the wrong side. You can see the guys are coming into the river. They're booting across over here. This is the confluence of Zeta Creek and the Pembina River. So guys are booting around in the river and, I don't know, cleaning their quads or something like that. Who knows? But that was another one that we reported and said, guys, there's quad issues down in this particular area. As a conservation group, we look for opportunities to do some stream side remediation. So there was a site on Dismal Creek that we went in and did some willow cuttings, basically in an effort to enhance some erosion. This used to be a bridge crossing, and there was quite a significant set of erosion there. So our folks went in and gathered cuttings and planted them along the side of the stream. And that was a reasonably successful effort. Particular season that we chose to do it was awfully dry. A lot of them didn't take root, but the idea is that we get some roots going in here, and that will help firm up the banks and prevent the erosion. Our DNA work was really interesting. We worked with some biologists and a student that was working on some master's work. Compared DNA from fish fin clips throughout the upper Pembina. And basically, this research work that was done had samples from Athabasca and the Peace in the Hay River. Our folks went out and gathered a bunch of new samples. And the samples that we collected on Rat Creek and Dismal Creek were very significantly similar to the ones from the Athabasca system. So Athabasca, basically this is showing that of all the fish, of the hundreds of fish that were caught, there were genetic markers that were very similar in the Athabasca and a distinct set for the Peace in the Hay. What was really interesting was as we peeled back the layer of the onion and went a little bit deeper and looked at the next level of DNA, we'll call it that, we found that there was a difference between the samples from Rat Creek and Dismal Creek. So Rat Creek, what we've got is older samples on the left, newer samples on the right. The Pembina River, Dismal Creek are very similar, and the Rat Creek from both before and after the sampling, the two sets of samples, really shows that there was distinct populations between Rat and Dismal. They're really not that far away. They're connected by the Pembina River. That's a little bit of a surprise. So we've got some resident fish probably in Rat Creek. And this is important because if we ever get to do some repopulation of fish, either by restocking or movement, that kind of thing, it's important to get fish that have similar DNA characteristics between from the target and the source. We also participated in another activity I mentioned a second ago about how the peace fish were a little bit different than the Athabasca fish. And basically, after some research, largely by this team, there's a geneticist and a few biologists that were working on a project to go and collect fish samples from the northern part of the province as well as the Athabasca and compare them. And they basically found that there's two different lineages of Arctic grayling, the Beringia and the Mahon. The Mahon are in the north and the Beringia are the ones in the south. The Beringia are a little bit more common throughout the system. The Mahon, though, you can't easily tell the difference by looking at the fish, but by photographing and taking some measurements and by looking at the DNA, you can tell the difference between these fish. There's ongoing research happening here, and eventually there may be a different management required around the Nahani fish. So on the right there, this is me. There's an interesting project. We hopped into a few different streams way up in the north, and we're collecting samples from the different lineages of Arctic grayling. We also did some environmental DNA work. So environmental DNA is where we can collect a water sample And then based on the analysis of that sample, we look at basically skin, excrement, mucus, other things, and detect if there's Arctic grilling in the water. So we did a project where, okay, let's skip the slide there. How's it useful? Basically, by taking a water sample, we can determine if there's Arctic grayling in that particular stretch of water, or if there had been in fairly recent period of time. And it's very efficient. You can go and sample a number of sites all in one day, just collect the water bottles, filter them, send them to the lab. You can cover a large area and compared to angling, which means that you have to go and spend several, sometimes hours at a site trying to catch fish. So it's more efficient, and it gives us some information about the range of where the Arctic grayling are. So we had a number of sites that we tested. A lot of those cooler sites up in the upper water, maybe too cold for Arctic grayling in the time of year that we were looking at, and basically very few Arctic grayling up in that part. But we did find quite a number of positive samples down in the mid of the upper Pembina range. There's a couple of oddballs too. Way down here in the southern part of the Pembina, there was a hit that we didn't expect because the water was a little bit warmer. Theory is that a fish had died and floated down, and some of his DNA was in the water there. And there was another one oddball up here on Paddy Creek. So some of these warmer streams still had some evidence of Arctic grayling in them. So that was kind of an interesting finding there as well. But overall, it was quite consistent with what we were expecting based on our angling. I got a few slides from the folks at environmental and protected areas. Over a number of years, starting in 2014, they had been doing some raft electrofishing on the main stem of the Pembina River, as well as targeted angling on a number of the tributaries. And there was an angling regulation freeze, so no angling allowed in those areas for starting about 10 years ago or so. So what we're looking for was, are these measures that we're taking, are we getting bigger fish? I'll talk about some of the indicators. The first one was bigger fish. So basically, in 2014, there was some medium-sized fish. 2020, they were getting a bit bigger. We've got a few of that class seem to move off into the larger fish size over here. And one more sample year that kind of flattened out a little bit. So we got a little bit of trending to, yes, we're getting some bigger fish. Another indicator was something called the Fish Sustainability Index. And basically, the biologists look at a number of factors and they say how threatened is this species in this particular area. So basically, level 4 and 5, or an index level of 4 and 5 are good. There's no real problem there, and we know that we had historic values in there that would have put us into a low risk range. We'd like to be in the moderate range, but at the beginning of the closure, their index showed that we were very, very high risk and close to exportation. After 3 years, we were getting a little bit better, but even after 6 years, we're still in the very high risk range. Part of the problem with some of this data was that there's only 3 or 4 years of sampling, 4 years, and 2 of them are a couple of pretty close years. Really hard to get long-term trends with so few data points. Are there fish in more places? So in 2014, 10 of the sites that were visited had fish caught at them. 2018, 11 sites, 14 sites in 2020. So they seem to be spreading out a little bit more in 2020. So that's another indicator of things are looking good. An additional indicator is based on the sustainability index, they're trying to look at is there recruitment, and very little recruitment across the years as well. One of the problems is that we're trying to get a 23, 24, 25, 23, 24 index as well. But due to forest fires that we talked a little bit about before and very high flood levels, they weren't able to get those that basically next data point to really come up with some trending information. So next steps, what's going to be happening? Within the club, we'll be looking at doing some more temperature monitoring video inventory. We want to collect some more DNA so that we can do some more extensive analysis before and after this 10-year sampling period. I talked to the Alberta Environment and Parks folks the other day, and their plan is to do some raft sampling this year. Some more angling, and we'll help them with that as well. DNA sampling, temperature monitoring. We basically split the temperature monitoring duties. We do about half of the sites each. And in 2026, there should be a report on the state of that watershed and some recommendations coming out then. We're really interested to know if there was a response in the fishing community around, and that's the overall community, whitefish and grayling and the other species, as we approach that 10-year. So by looking at some DNA and figuring out basically the lifespan of these fish is a few years, you can tell a little bit of information about where they've moved, how far they've stretched out, based on familial characteristics and so on. So that's a sort of a focus over the next year, and there's a little bit of a grant application in progress there to help in that effort. So we'll be looking at doing some more sampling and DNA work as well. One thing we'd really like to get at is a feasibility assessment around Arctic grayling translocation. So there are technologies that would allow us to there's one called stream-side incubation. So basically you gather some eggs and gametes and you put them together, but instead of raising the fish in a hatchery, we would basically let them incubate at the stream side so that when they hatch out, they imprint right onto the stream that they're in and increase the likelihood of being able to come back and do spawning and so on. They've done some of this work in Alberta for rainbow trout, cutthroat trout, and there are other jurisdictions in the States that have done it with Arctic grayling. So we're hoping that at the end of this assessment phase, we'll be able to really understand if we can do some of this translocation with the Arctic grayling. That's the ultimate goal. I'd like to thank many volunteers from the Northern Lights Fly Fishers and the Alberta Conservation Association for their financial support with this project. And that's my last slide for today.
Thank you very much, Ken. Shayla, do you have any questions to share with us?
Yeah, so Sarah asked a question. How did the 2023 fires in the Pembina watershed affect your stream temperature program and results?
Well, it was interesting. 2023 had the fires, and as part of the overall program, so that wide fire area covered a large part of the middle part of our study area. So we weren't able to get our temperature loggers into a little bit later that year. And our drone program, we weren't able to fly over these or even near these fire protected areas. So the fires were a big impact. But shortly after the fires, there was a lot of flooding. So if you remember back, there was tons of flooding. In fact, we lost a bridge on Rat Creek and we lost a bridge on Dismal Creek. 2 bridges washed out completely because of the amount of water that came down. So that caused a lot of sedimentation changes in the rivers. It caused cooler water coming down. You might have expected the fires would cause a lot of higher temperatures. In actuality, because they were followed by those floods, we got a sort of a mixed bag of results. It was interesting and requires a little bit more research. Our chapter does meetings every couple of weeks, and we've got one coming up that anybody will be welcome to attend. And there's a fellow from UBC talking about UBC, a university in British Columbia. I'm not sure if it's UBC. We'll be talking about the impact of forest fires and stream population. So that'd be an interesting talk coming up.
Awesome, thank you. Don't think there are any more questions in the chat, but I'll put a link in the chat right now to the Northern Lights Fly Fishers.
Thank you.
so that if anyone wants to find more information about the Arctic grayling work or about that meeting, they can.
Thank you.
Yeah, of course. Thank you for your presentation.
Yeah, thank you, Ken. That was great. Sarah, would you like to jump in?
Yeah, thank you everyone so much for joining this webinar. I just wanted to, I have a slide here to share. Check out our YouTube channel, check out our website, and if you want to get in contact, my contact information there. And I think Shayla's putting all that in the chat. So we really appreciate your time joining us today for these 2 hours. And I hope you all have a wonderful rest of your day. And thank you, Ken, Lauren, Theo, and Jennifer for your wonderful presentations.